right, welcome everyone and good evening. My name is Mary Kinney. I am a board member and publicity chair for the Portland chapter of the Native Plant Society of Oregon. NPSO promotes the enjoyment, study, and protection of Oregon's native plants. I'd like to especially welcome any new members tonight. As part of our mission, we present a speaker each month on timely topics of interest to our members. Our speaker tonight is Mitch Cruzan, presenting the saga of the Slender false Brome invasion and lessons for the future of our native plants. Before we get to that, let's review some Zoom etiquette. Your audio and video should be turned off during the program. After the presentation, you'll be able to unmute and turn cameras on. That will be the perfect time for new members to introduce themselves and connect with each other virtually. If you have a question during the program, you can click the chat button at the bottom of your screen and type it into the chat box. We'll address questions after the presentation. You may also notice a closed captioning feature. You can enable it whenever you'd like. First, just a couple quick announcements from our chapter vice president, Willow, who's in the audience today. September and October are public land stewardship months, and PSO members are encouraged to attend native plant restoration work parties that are listed in our chapter newsletters in September, October. And if you're not a member, you can look for events in your local watershed council or city parks or regional parks. Volunteers power regular weed removal projects, especially to return native plants to their original habitat. So it's really crucial for us to volunteer during this time of year and any time of year. Also exciting news, save the date for the 2025 Native Plant Society of Oregon annual meeting. The Portland chapter is hosting the biggest gathering of the year on the weekend of May 16th through the 18th at the Columbia River Gorge Best Western plus Hood River Inn. Be on the lookout for more details in the October 1st state bulletin to reserve a room at a discounted rate for early birds. You must be a member to attend, so make sure that you join NPSO soon and go to the state website at npsoregon.org. All right, now it's time to introduce our speaker. Mitch Cruzan is a professor of biology at Portland State University. He received a BA and MA from California State University, Fullerton, and his PhD from SUNY Stony Brook, and served as a postdoc at University of Toronto and University of Georgia. His first faculty position was at the University of Tennessee, Knoxville, and he has been a faculty member at Portland State since 2002. He teaches upper division courses, including evolution, evolutionary genomics, conservation genetics, plant reproduction, and plant evolutionary biology. In his research, he utilizes ecological and molecular genetic and genomic techniques to address questions in plant ecology and evolutionary biology. His research includes the evolutionary consequences of mutation accumulation in plants, as well as studies in plant reproduction, hybridization, invasive species, phylogeography, and dispersal. He is the author on over 80 scientific articles, an associate editor for 2 leading journals in his field, and is an author of an advanced textbook titled Evolutionary Biology, A Plant Perspective. He is currently serving as the director of Portland State University Herbarium, chair of PSU's Greenhouse Oversight Committee, and a member of the Faculty Senate Budget Committee. Welcome, Mitch.
Thank you, Mary, for the nice introduction. And thank you, Cheryl, and anybody else who was involved in letting me tell you guys about what we do tonight. So I'll just go ahead and start sharing the screen. Hopefully that works. And is that working for everybody?
Yep, just got to full screen. Looks great.
And you can hear me?
Perfect.
Okay. I'm just going to mute my video. Okay. All right. So we've been working on the invasive false brome for, God, I was trying to decide when it was we actually started. I think it was soon after I got here, because soon after I arrived, I recruited a graduate student, Alisa Ramakrishnan um, from Utah, and she was really interested in invasive grasses. She had previously worked on sheetgrass and was looking for a new system, and she wanted something that was a perennial and was diploid. So doing genetics on things that are tetraploid and polyploid is a little bit more complicated. So we cast around, and my first suggestion to her was because I had just come from Knoxville, Tennessee, and there Japanese grass is a big problem. That one was introduced, and all the instruction stories are kind of interesting. From what they know, that one was introduced by packing material on a shipment from somewhere in Asia that somebody basically unpacked in Knoxville, and the seeds escaped, and now that's a major pest. And it's an annual major pest in all over the East Coast, and I hope it never comes here. I think it would thrive here. I hope it never comes here. It's a real problem, and apparently the seed bank lasts, oh, I don't know, years and years and years, and so it's pretty difficult to get rid of. Um, But then Elisa was the one who actually identified the slender false brome as a good candidate to work with. And so what we did then was to start in investigating where it came from, and how did it get in Oregon? Where else has it been introduced? And so in asking those questions, we basically started developing a whole story. And I think something like, we worked on it a good 10, 15 years. And so 8 or 10 publications later, we've put together, I think, a pretty complete story of this plant and how it has adapted to the Oregon environment. So species introduction in general is uh, uh, a problem worldwide. And the interesting thing to me is that generally these introductions are not accidental. I mean, there's some that are accidental. Like I think the green crab on the west coast of Oregon and Washington, I think that was accidental because it was from ballast that was released from ships that has now spread up and down the coast. But other introductions were intentional, and I think one of the most interesting ones to me is the European starling, and in that case, the starling. So going back to I guess it was the early 1900s, maybe late 1800s, there was a group in New York City, a society that their goal was to introduce all of the birds from the Shakespeare plays into the New World. And the starling, of course, was one of the targets. It took them at least 3 tries. And finally, in the third try, somebody brought in something like 60 birds and released them in Central Park. And you know what has happened since then. Um, there's kind of an unconfirmed report that there was also an attempt to introduce the starling into Portland, Oregon, but that one did not take. So, people thought that moving species around was not a problem at all. And as you'll see with the false brome, that's a similar kind of story. But realize, and what I try to tell my students is that when we think about invasive species, we often think of them in a real negative connotation. We're frustrated with them. They're causing loss of of other species or native species, of rare, they're a threat to rare species. They're disrupting our ecosystems. Um, and but what I try to tell my students is that understand that the success rate of introduced species is extremely low. And there's this thing called the tens rule that 1 in 10 introduced species will actually become naturalized. And by naturalized, I mean that it can maintain its population through reproduction. And I think a good example of this is foxglove. And I'm sure all of you know foxglove. And it's actually a very pretty plant. And it's not an aggressive invasive. What I say, like to say is that it plays nice with other plants. It doesn't try to take over. But then 1 in 10 of those that become naturalized become what I like to call aggressive species. And false brome is one of these aggressive species. So the fact that such a small fraction of introduced plants actually become successful to the point they're aggressive means that, in a sense, we should kind of like maybe respect these guys a little bit, and maybe we can learn something from them. And that's kind of my goal today, tonight, is to talk about what we might be able to learn from these successfully aggressive invasive species and how we can apply that in ways that will actually help our native plants. So can invasive species serve as models for challenges of climate change? And to emphasize that point, understand that usually when plants and other species are introduced to a new region, it's not a perfect match for where they came from. And so there's a certain amount of adaptation that has to take place in the new environment. And so the question then is how they become adapted so quickly to novel conditions. And particularly in the case of Oregon, there's really not a climate, there's not a we're like a cool Mediterranean climate. And there's not a matching climate for us in Europe. And so what was introduced from Europe was maritime, um, tropical, or like not tropical, but subtropical Mediterranean, but not a temperate Mediterranean climate like we have here. So in the late 1800s, and it was around 1888, 1889, there was this cushiony scale insect uh, was a major threat in California, nearly destroyed the citrus industry in California. And one of the entomologists working with the USDA realized that it had been introduced from Australia. And so they went to Australia to find out what was the natural predator of this cushiony scale insect. And it turned out to be the Vidalia beetle, which is sometimes called the Australian ladybird beetle, or just or sometimes the Vidalia ladybird beetle. But it's not the same as our native ladybird beetles. It's different. And currently it's often used in um, as a biological control even in North America, because people think, well, even though it's been introduced, it's still a beneficial insect. And it was, and it basically solved the problem. The success of that experiment got people thinking, and they thought, well, I wonder what other species we can introduce to actually make our rangelands and our forest and our everything better than it is. And so there was a USDA program established for the and currently it's called the Plant Introduction Program. It's been gone under several different names, and now actually it's kind of merged with the germplasm um, preservation programs as well. But still today, there are plant introduction program sites, one of which is in Washington State, and I just spoke with them a few weeks back because we're interested in starting to work on false brome again. And they still have these collections from all over the world of false brome and literally thousands of other species that they maintain. Um, and so what happened then, and let me just check, is that both in Eugene and in Corvallis, test gardens were established for the false brome. And so what they did was they were trying to basically improve rangelands to basically have food for cattle, to make more hamburgers, I guess. So that and they had the best intentions in mind. Um, it was a program that um, was intended to actually benefit the state, okay, economically, and then they didn't really think about the implications of what they're doing. From what we can, well, actually, I just want to point out, it actually is a pretty beautiful grass when you look at it. It's got these sort of elegant sort of um, leaves that drape down. It's very soft. It has the hairs on the leaves are long and kind of lanulate, and so it feels very soft to the touch. And you can distinguish it from Columbia brome, which looks very similar, by the soft touch when you actually touch the leaves. If you find a grass that you might think is false brome, that's a good way to identify it. And they also the inflorescence is the fruiting stem is also sort of, the seeds are very loose and kind of also sort of drooping. And it doesn't shatter, it doesn't lose its seeds right away. It holds on to them pretty well. It's easy to propagate by tillers. It's a bunch grass. It's diploid. It's wind pollinated. Um, it's we think mammal seed dispersal, that animals pick up the seeds, and probably one of the biggest dispersal agent for this plant is humans and their pets. And so you can find it on road pullouts along the highways going out from the valley up into the Cascades. And what I think happens is that people let their dog out to pee and get some seeds on it, and then they stop at the next place, and the seeds get dispersed. And a single seed can establish a new population because it's self-fertile. Okay. So at one point we tried I had some students out at MacGyver Park, and we were trying to figure out, well, how does it move around? Does it catch on the clothing? And I had the students, they were like, had different kinds of jeans. One person had corduroy pants, and they're walking up because there's really dense populations down near the river there, and they're seeing if they get any seeds on their clothing. Turns out that the seeds were actually on their shoes, both in the laces, and then it was kind of muddy. So when they were scraping the mud off the bottom of their shoes, there was lots of seeds in there. So it's very easy to move this particular plant around. No, it's doing this. Yeah, and it does have a fungal endophyte. And I don't know if you guys know Biddy Roy at University of Oregon, but her lab has studied that quite a bit. Turns out that the endophyte probably was what made the plant really not palatable for the cattle, which is the reason that they apparently abandoned this effort. We've worked on the mycorrhizal fungi of this plant. It does seem to have different mycorrhizae than native plants, and it might be a bit of what people call an eco-engineer, that basically makes the habitat less hospitable to other plants by favoring mycorrhizae that other plants don't um, particularly uh, are useful to them, or beneficial for them. And again, these seeds from all over the world, including places you can't get anymore, Afghanistan, Iran, Iraq, all over Europe, are available and are maintained. Once in a while, they grow them out periodically to generate more seed to make sure they have viable seeds from all these collections. And there are, for me, for my stuff, genomic resources, and now we actually have a complete nuclear genome for this particular species of Brachypodium sylvaticum, because it turns out that Brachypodium is sort of a bridge genus between a couple of crop grasses. And so there's been a lot of interest in developing genomic resources with this plant. So where did it come from? Well, its native range is primarily Europe, Eurasia, mostly Western Eurasia. Um, and then it's been introduced from there into the Western North America. And this particular slide is only showing some of the original introductions. But now we know that it's been introduced several places in the eastern US and actually around the world. If you go to iNaturalist and you type in slender false brome, you'll come up with a lot of different locations that it has spread. So it seems to be a very good invasive plant. Um, I should mention at this point, there's actually, I'm going to talk about two introductions here in Oregon, and there's an independent introduction down in California. And that one in California is really interesting because in that particular case, it was somebody who had intentionally brought the plant from Italy, and it's a region that's just west of Stanford University and the foothills there. And I was actually there at the address and was collecting leaf material for genetic work. And somebody, a woman come up from the house, not from the house that it came from, but the one below it on the hill. And she immediately knew what I was doing, that I was working on false brome. We had a good conversation. And she said, yeah, that guy, he introduced it and it's spreading around here and he doesn't care. So um, yeah, and it was all over her property. So it was just, coming downhill basically. Um, so that particular case was a little bit more unusual because it was a single source introduction. And what I'll talk about in Oregon were multiple sources. Because in Oregon, when they planted those test gardens, they put together uh, plants from all over Western Europe and the Middle East, all together growing in the garden, as far as we can tell. We don't know exactly what was planted. We don't have any records. But we do know that what's spreading in Oregon is basically a hybrid between uh, there's components of plants from England, France, Spain, Italy, and Greece in the actual this sort of like this hybrid aggressive weed that we have. So this is the history of herbarium records, and notice that the first collections were made in Eugene in 1939. We think the gardens were actually started quite a bit earlier than that, probably in the 1920s, but again, we don't know for sure. Then by 1966, they were starting to see it in the Corvallis area, and I think what happened is that they either abandoned the gardens or maybe seedlings propped up around the gardens and they didn't notice. Uh, and notice that by 1989, what you're seeing is that a lot of collections around Corvallis, and Corvallis in particular. If you've ever been to the McDonald Dunn experimental forests. There's areas of McDonald Dunn forests that are just, the understory is almost a complete monoculture of false brome. And it's basically outcompeted the sword ferns, salal, snowberry, all the other stuff. You see, there was one place we were at one point and we stopped and I looked, and as far as I could see was just false brome on the understory. I think a lot of that has to do with the fact that those particular forests have been used for trying different cutting techniques. And so there's been a lot of soil disturbance which I'll get to in a bit. So there's 89. It was after about 1990 that then it really started to spread. And so this is 2003 herbarium collections based upon collections at OSU, and I think that Elisa used the Pacific Northwest Consortium of Herbaria to get these data. And then this I pulled from iNaturalist just a week or so ago. And so notice that if you look within our particular region, it's really concentrated in the Willamette Valley. And as of about 2010, it was pretty much restricted to the Willamette Valley and then some down in the Southern Oregon, but it hadn't jumped across the Columbia River yet. And so now if we skip to the present, and this was just, like I said, a week or so ago, we actually have collections up over the Canadian border. It's, again, a lot of density just here in Oregon, and then it's invading into the redwood forest, actually, State Redwood Park in Northern California. Here's the California introduction, and notice that it has not spread nearly as much as the Oregon introductions have. These two independent introductions. Oops, sorry. So one of the things that we did was to say, well, we can run this program to basically ask the question, well, we know where it grows, but where could it grow? And so what you do is you use these geographical information system layers that have provided the climatic variables or soil variables, all sorts of things you can put in there. And it's kind of a little bit like AI. You train it and say, this is where it's growing now. So where could it grow? And this is from, I think, 2010, or no, actually, this is from 2018, I think. That's what we had at the time. And this is just based on our barium record and not iNaturalist. And so that's the points we use to train it. All of this red area is potential habitat for false brome. So all the way down into Southern Oregon, and notice Northern California, along the slope of the Sierra Nevada, it has a lot of potential. So this particular invasion is actually pretty interesting for us to study because it's in the process of expanding its range. The whole invasion is not nearly over at all. If you look at some of the eastern invasions like kudzu, or even like English ivy here in Oregon, it seems like it's probably, extended where it's going to go in terms of geographically, not all of the local locations, but geographically. So it's a pretty ideal system for studying this kind of stuff. Now, I don't want to overload you with oh, here's genomic stuff. Okay, sorry. So I want to give you a little bit of this idea, and what I want you to focus on are the colors. And what this represents are what we call these genetic clusters that correspond to basically the genotype, like a genetic fingerprint. And notice the genetic fingerprints from the native range are all these kind of blues and yellows. And then if we come into Oregon, we get these oranges and purples. And what this told us is that what we have growing here in Oregon, spreading here in Oregon, is a combination of the genotypes that came from Europe. And particularly, as I mentioned, it was England, France, Spain, much less of Italy, and a little bit of Greece. Notice that the California invasion is its own color over here, and that probably corresponds to this blue that's also found in Europe. So that was a single source, whereas this represents a lot of hybridization that have gone on. We also have looked at how plants have changed since they've been introduced. Since we knew where it came from, we can compare a lot of physiological characteristics between the introduced range, the invasive range, and the native range. And in particular, we were interested in this because we have a much longer drought period than much of the native range, particularly France and England. So notice that all four of these represent different physiological characteristics that are drought adapted. And so these arrows are giving us an indication that increased drought adaptation in Oregon, increased drought adaptation in Oregon, going down, increased drought adaptation in Oregon. And so this is what we basically found that once this plant was introduced, the reason probably that it took a while before it spread was that it was undergoing this adaptation process. There was a lot of strong selection on these plants, a lot of them not doing well. Some of them were, the ones that were did better and then spread their seeds, okay? The ones that emerged from this whole process were these aggressive hybrids. We've also looked at the ecology of the spread. And so, and this work was done by Gina Marchini, who is now the owner of the Banshee Shop, which is a retro dress shop in Oregon. I just want to put a shout out for her. And then this work was done by Laura Taylor, who works with the Multnomah Water District now, and she did her master's on the ecology of the spread. And so what we did with a whole bunch of undergraduate students was to ask the question, how does the places where false brome is growing and really creating a monoculture different than the places where it's not growing, that are basically, you get these dense patches of false brome and then just 10 feet away, native vegetation. How is it different? And this work was done at MacIver State Park. And notice that we have indications of disturbance of some kind. So the litter was removed, okay? Where false brome is present, a lot less litter depth, a lot less litter mass. Understory height, shrubs and stuff, a lot less shrubs in the area, a lot less density of native understory shrubs that you would expect to find there. So apparently they were either destroyed in the disturbance or pushed out by the false brome. And then also soil compaction. And that was a huge one for me. Realized that there was, maybe they were doing some work there that caused the increased soil. And it's not just like the soil compaction, but I think the soil compaction is a symptom of disturbance. And so the lesson here is that if we don't disturb this nice thick duff in the understory, we can probably prevent a lot of invasion of natural habitats. The other thing that Laura Taylor did was to look at the different conditions that she, where she looked at like disturbing, like making mineral soil. She just kind of tilled it. She had a control. She had some mulch on there. And what she generally found was that when you do these disturbances, you basically invite the plant in. Okay. One of the real advantages, and there was Columbia brome growing nearby, but what she observed is that the false brome seedlings were a lot more dense. And a lot of it was because false brome makes a lot more seedlings. And so the more seeds it makes, the higher density of seedlings that you get in the disturbed areas. Another thing we were interested in was how it spreads. And this is work by Elisa Ramakrishnan, who did her PhD with us. And she wanted to sample along roads, and this is Highway 20, going up into the Cascades, and she sampled some side roads, and all of these little triangles represent basically populations of false brome, and some of these are just pullouts, okay? And so what she wanted to ask was, when you get these new little populations out here, where are they coming from? Is it like, are they doing like a hopscotch, one after the other? Or are they moving like longer distances along the road? Um, and this represents basically what she found, which is that this population, which was the largest and densest and had the biggest individuals and genetically had the greatest heterozygosity, the most vigorous, um, uh, is the major source for a lot of these new little populations. So the thickness of these arrows indicate basically how many plants in these new populations could be assigned as coming from this M6 population. And so this is interesting because back in the 1980s, there was a very influential paper written that basically said if you want to stop an invasion of a plant, you should go out to the edge and you could stamp out these little populations out here. But this result tells us a completely different story, that this is like, I mean, I kind of think of like a war. This is, the source of resources coming, and when anything gets, even like in these cases here, what we called M3 and M1, those populations, they disappeared. But I'll bet if we go back there today, they've reappeared and they probably came from this guy. So it's a recurring thing. If you really want to control this thing, you go to populations like this and you take them out. Not the little weak ones, the little weak ones on the edge are maybe not contributing so much to the invasion. The other thing that happens with genetics, and one of the sort of the puzzles of invasions, and particularly with a self-compatible, self-fertile plant like false brome, is that we know that when the things self-fertilize, that they're less vigorous. And this is just sort of a classic experiment with this white clover. These are the inbred plants, and these are the outbred plants. And we call this inbreeding depression. We call this due by genetic load, which is basically deleterious mutations. And they're generally, if you remember some genetics, they're generally recessive, so they're not expressed when the plant's heterozygous at that locus. But when they inbreed, they're more likely to be homozygous, and so that's when you get the inbreeding depression. Okay, why? And imagine that the process of invasion of false brome. Why is it not suffering inbreeding depression if these populations are being colonized by single seeds and they're self-fertilizing? I mean, that's kind of a puzzle, and people have wondered about this. Like when there's an introduction, it should be a little tiny population. Inbreeding might slow it down. This is the only graph I'm going to show you. I guess the other one was a graph, but this is the only like complicated graph. But it's actually pretty simple to understand. This is fitness, which is basically plant vigor, including seed viability. And notice that down here we have a population right near Corvallis. We have populations a little further from Corvallis, a little further from Corvallis, and then we start getting out to the range edge, the expanding range edge. Okay. And what we see along this gradient is an increase in the vigor of plants. Notice down here, these light bars are self-fertilized. They're not doing so well compared to the outbred ones. But once we start getting out here, the self and outbred ones are doing about the same. Okay. And so what this tells us is that during the process of range expansion, these plants are doing what we call purging genetic load. They're losing their deleterious mutations. So they're no longer suffering from inbreeding. Back here at the beginning near Corvallis, there's a lot of inbreeding depression. Okay. Now this is a little bit alarming because it means that the most vigorous plants at the range edge are producing more seeds. They're going further. And like I said, what we've seen is that the actual invasion seems to be accelerating. And so just kind of as sort of a diagram of what we think is happening, here's our native range. We get a lot of hybridization once it's introduced into Oregon. There's really strong selection, and we go through what's called a population bottleneck, during which there's inbreeding, and we get all that genetic load. But then as they start spreading, they lose that through selection as they spread. And so what we're seeing now is a very aggressive plant at the range edges that's, like I said, they're already in Canada. So what about native plants then? What does this help us with native plants? Well, there's this sort of little, idea within climate change research. We've done quite a bit with looking at seed dispersal, particularly for plants down in the Medford region, looking at how well can they move from one habitat patch to another. And so there's this idea, well, either they're going to stay in place and evolve, or they're going to have to move. Okay. And so the our research sort of spans the whole sort of moving part and the evolving part in terms of the persistence of native plants. And just to kind of show you the challenges that plants have to deal with, this is a, a land use map for the Rogue River Valley. And, um, and so the red are basically urbanized areas. The dark brown are cultivated crops. And then this yellow is all hay pastures. And so if you know this area, here's the Table Rocks here and here. Those are beautiful habitat for plants. Notice what's in between them. So what we've done, and I was going to show a picture of the Willamette Valley, but it's even much worse. Okay, what we've done, we've actually made it much more of a challenge for plants to move around from one habitat patch to another because of our land modification. Now, one thing that we've learned in this, and this is one of the plants we've worked with in Southern Oregon, but it's a beautiful little plant. It's one of my favorite. Aplytritis congesta, the sea blush, bumblebee pollinated. It's an annual, and it's just gravity dispersed. And then you ask the question, well, what if the seeds are just falling out onto the ground, how do they actually get from one place to the next? And so based upon our research, that what we've learned is that, let's do this first, is that it's the migration corridors of the plants line up pretty well with wildlife corridors. And so a lot of the seed movement for many plants that don't have specialized dispersal mechanism is probably seeds getting caught up in the hooves and the fur of elk, deer, and other large mammals that are moving around from one place to another. And so what we've learned is that that you can actually get a lot of connectivity from one place to another. And it's not just based upon distance, but it's based upon things like geographic barriers, rivers, riparian areas, contribute, sort of redirect the paths that seeds take from one place to another. So one thing to think about then is that in terms of movement, maintaining wildlife corridors not only benefits these the wildlife, but also the plants, because they're carrying seeds with them. So the other thing I want to put out there, and this is maybe a bit provocative, but I think all of us are very much in love with the biological diversity that we think about as different taxa, different species, right? Um, my partner Carol and I have put together a native plant gardens in our front yard, in our backyard. We've tried to make different kinds of habitats. Stream, we've got rock gardens. I've got a bog with Darlingtonia in it. And I think Carol's logged something over 200 different species that we've gathered together. So we love these guys, right? On the other hand, if we think about the history of all of the species that are here in Oregon now, that they haven't been here for very long. And not only that, if you look back over the last, say, few million years, we see these cycles of glaciation where the distribution of different species are moving north for a short period and then south into glacial refugia for a long period, and then diverging into new varieties, subspecies, maybe species. And then when they move north again, they contact each other and they interbreed, they hybridize. And so if we want to think about long-term preservation of our biological diversity, maybe more than thinking about these species as static entities, maybe we should think about them more about what their history is. We know one of the great lessons I think we've learned from the last few decades of the sort of whole, DNA sequencing revolution is that almost everything out there has a history of hybridization, including us. And that hybridization we think has been important. And that idea goes back all the way to the 1930s when the evolutionary biologists were like Ernst Mayr was saying, hybridization is not important. Edward Anderson, a plant evolutionary biologist, and Ledger Stebbins were saying, oh, hybridization, they called it uh, uh, something that would promote adaptation because they understood that in plants we see a lot of hybridization and that is important. They didn't really understand the full range of hybridization of all these different species, but it happens. And so things like that people are starting to do now, assisted migration, assisted gene flow, these in some ways are mimicking the history of species. And so maybe it's more about preserving evolutionary processes rather than treating these taxonomic species and varieties as just static entities. Because one thing that we know, one thing that we learned that Darwin taught us, that he viewed all species as having a history and a future, that things have evolved, are continuing to evolve, and will evolve in the future. And some of that evolution has to do with hybridization. And I just wanted to show, because the work I've talked about has gone back a couple of decades. And then here's some of the grad student and undergraduate student groups. These guys worked on false brome, many of them. We worked with Biddy Roy at University of Oregon. We got seeds from Kew Gardens and other places. And then some years later, the work with seed dispersal in Southern Oregon, that was collaborative with folks from University of Oregon, Duke University, University of Colorado, um, and included a whole different group of folks, and then postdoc, graduate students, undergraduates, and then my current group. And we're actually working on, in addition to well, I guess there's one person working on false brome, but mostly we're looking at how plants evolve in terms of their accumulation of mutations. That's a big push. Also looking at what's called developmental selection, which is things like pollen tube growth and seed development and how that can actually contribute to selection and adaptation to plants. Um, Zichi's working on hybridization in ranunculus ostroorganus, which is restricted to Jackson County. It's just really range limited, and understanding, how it might persist and how genes are moving between it and the Western buttercup, Ranunculus occidentalis. We're doing some work with assisted gene flow in Eastern Oregon, working with some BLM managers out there for basically trying to improve drought tolerance of some of the grass species they use for um, restoration projects after fire. Uh, we have an undergraduate who is using herbarium records to look at how flowering times have changed pre-1970 versus post-1970. And it's turning into a really interesting story. The effects of climate change are not consistent across the Western US. Some particular kind of ecoregions are being impacted much more than others. And then again, and our undergraduate Annabelle is looking at the genomics of genetic load in false Brome. And that's all I have. So I'm going to unshare. And I hope that wasn't too overly technical.
Thank you so much. Yeah, that was super interesting. I think it's an interesting way to frame, what we're doing with our native plants and how we're kind of approaching invasive species as well. There are some questions in the chat. I can ask them for you. And then for folks who do have questions, please put it in the chat. I will read them for you. Caitlin says, I've heard a lot of emphasis on sourcing seeds of populations that are specifically growing in the area, aka ecotypes. What are your thoughts on this emphasis, as it seems to go against your emphasis on genetic diversity?
Yeah, I think one thing that we need to appreciate first off is that particularly in plants, um, usually seed and pollen dispersal is really local. And so in general, what we find is that individual plant populations are pretty inbred, meaning that if you go out and collect seeds from a single area, um, not always, but often you'll get, low seed germination and low vigor. If you were to take um, pollen from a different, location that not even that far away, maybe a few kilometers, and then move it to an area and pollinate flowers, you would probably get more vigorous seed because the populations tend to just randomly become what we call fixed for different deleterious mutations. And so when we do some gene flow, we can overcome a lot of that genetic load. But more broadly now, I think we have to think about, with climate change, maybe doing a little things a little more adventurous and moving seeds and plants from areas that are, you know, further south in areas that are subjected to greater drought. And that's what the assisted gene flow tactic is trying to do. In particular in Eastern Oregon, we've identified areas in Eastern Sierra Nevada, just in the valley, where there's more severe drought than in Eastern Oregon. And so we're trying to do assisted gene flow from those populations to up here and recognize that and I know there's this thought that, you want it to be just what's local, but given what we're facing with climate change, I think we really can't afford to do that anymore. We need to think more broadly. That's just my opinion.
Yeah, thanks. Shannon asks, are there any of the papers mentioned in this presentation available to read anywhere, or where can we find them?
Yes, actually, there's one in particular if you want to know more about the false Brome story. I have a paper in bioscience, and I could actually put a link to that in the chat.
Yeah, that'd be wonderful.
Um,
Yeah, and while we're waiting, if there's any other further questions, happy to ask them.
Interesting in your genetic analysis of the bottleneck and then subsequent adaptation and increase in diversity. That explains a phenomenon I noticed in my reviews of impacts of biological control. It's quite frequent that the initial introduction of biocontrol agent is unsuccessful, and that includes your example of the Cotney-Cushing scale, which the original introduction to California was from the native population in Australia, and that failed. Every introduction of that failed. What was finally successful was an introduction from an invasive population discovered in New Zealand, and that succeeded almost immediately and was the source of the control. And most of the biocontrol agents experience has been that to collect propagules from an invading population in a new land and introducing that into agricultural systems, those are successful.
mhm.
And your bottleneck is a good explanation of that phenomenon.
Mhm. Yeah, when
And
I've looked at the literature, I think it's much more common for invasive species to have come from multiple introductions from different geographic regions rather than a single source, like the case of the false brome in California. The real successful ones, I think, tend to have high genetic diversity in the invaded range, which has to do with um, the uh, uh, the way that it's been introduced from multiple locations. But yeah, the population bottleneck, which seems to have happened in this case, yeah, that's that's probably common as well.
Cami asked, in the face of climate change, what tools are you using to identify those drier, warmer locales for seed collection?
Yeah, so we can use there's the whole set of GIS layers provided by WorldClim, and then also this organization called Prism, that's based in Oregon State University, and you can basically look at precipitation levels and um, drought indices in different regions and identify areas that appear to be more severely dry than they are here in Oregon. So that's how we identified the eastern region of just east of the Sierra Nevada.
Yeah, cool. Few more questions. Caroline asked, now that it's known to be invasives, are there restrictions being employed on the false brome?
Well, there's a whole resource managers are very aware of it now, and there's a lot of work to try to control it, including spraying and um, and so I think what people have realized is that it's probably not possible to eradicate it or even stop it, but rather we can probably slow it down. And so what I was suggesting is to go after these big source populations,
Yeah.
and that might be more effective slowing it down than going after the little populations. I understand why the resource managers want to go after the little populations because it's a lot less effort, but yeah.
Definitely.
And the little populations become big populations eventually. So
Yes, that's true too. Yeah.
Yeah. Yeah, for sure. Haley asked, kind of on the opposite end of the spectrum, if our natives have become invasive in other countries, should we be asking them for seeds to bring those strains back to see if they perform better here in their native range?
Yeah, I know there's a number of cases of not nearly as many as the European sources, and but there are cases of our native plants becoming invasive elsewhere. I know that uh, I'm blanking on the genus that touched me now, jewelweed, is
Yeah.
in Germany. Um, I don't think it's super aggressive there, but it's at least, introduced and invasive. So and there's other cases, but the idea is that in Europe, the many more plants have been basically selected to adapt to human habitation a lot longer period of time, and that maybe there's sort of more plants that have the invasion potential. So think about all of the mustards, Brassica campestris, nigra, yeah, and other species. They've come, I think, many, many more from Europe to the US than vice versa.
Yeah. And then this is the last question I have, unless folks want to get their last ones in. Kristen asks, does the forest mastication as a pre-fire treatment have an impact on disturbing and reducing leaf litter, duff, and density, and potentially spreading slender false brome?
What was the first part of that question?
Yeah, so like if you're doing like forest mastication as part of like a pre-fire treatment, does that have an impact on that leaf litter kind of disturbance that can invite the slender false brome to come in?
Yeah, it's a good question. I know at one point Vidi was really interested in the effects of controlled fires on false brome. And I can't say for sure. I'm thinking that she found it was not effective at preventing its spread. But I'd have to look up that one up. So that's a little bit out of my area to answer. So
Yeah,
but
no problem.
but I think generally disturbing the duff layer in the forest is going to invite things to invade. And sometimes it happens naturally with tip ups, tree falls. But maybe we should all stay on the trail.
Certainly. Certainly we can all take that away from this too. Yeah. Well, I don't see any further questions. I would just really like to thank you on behalf of the Native Plant Society of Oregon and our chapter for just this really informative presentation. I think it was really illuminating.